Hello, everyone, and welcome to my dissertation defense. My name is Nazia Tabassum, and I'm a PhD student in the Department of Electrical and Computer Engineering at the University of Virginia. My advisor is Dr. Scott Acton, PI of the Virginia Image and Video Analysis Lab. The topic of my dissertation is image analysis for describing meningeal lymphatic vessel morphology. What are the meningeal lymphatic vessels? The lymphatic system is part of our immune system. Lymphatic vessels carry lymph throughout the body. Meningeal lymphatic vessels are a type of lymphatic vessel that is unique to the central nervous system. These vessels were recently discovered in a thin layer encasing our brain, called the meninges. The discovery was made at UVA in 2014, so much of their function is still unknown. Immune cells and wastes are drained through the lymphatic vessels from the brain, as shown on the figure in the left, through the green lymphatic vessels. Here is an image of lymphatic vessels stained in green, showing their placement on the skull from the back. In 2018, researchers from UVA, including the Viva lab, found that as we age, our lymphatic vessels thin and don't adequately drain waste. The accumulated waste leads to development of plaques, which is a common precursor for Alzheimer's disease. And Alzheimer's disease, as you probably know, is a neurodegenerative disease that results in the loss of memory. So the ultimate question is, can we treat these diseases by studying the structure of the meningeal lymphatic vessels? The figure on the right shows first a young brain with thicker vessels and adequate lymphatic flow. The effects of aging reduces lymphatic flow in the middle panel, and a potential treatment is to increase the lymphatic vessel size to reduce waste buildup. As the meningeal lymphatic vasculature, or MLV, and its role was only more recently discovered, there are no current image processing and analysis tools developed for analyzing these images. Thus, there are many open areas for investigation. But first and foremost, we need a method for segmenting these vessels or isolating them from their cluttered background. We then want to analyze the error that occurs in manual segmentation, as this is the state-of-the-art method for segmentation. Since the manual annotation is shown to have high error rates, we create a synthetic data model in order to provide unimpeachable ground truth. Although the data available is limited, we explore the frontier of deep learning for vessel segmentation, which shows us promising results for future work. And finally, with segmentation in hand, bioinformatics can be calculated on the vessels, which will provide us with structural information. There are 5 contributions or objectives of this dissertation, fulfilling the needs discussed in the previous slide. So the first is to create an automated segmentation method called Lymphy to segment the meningeal lymphatic vessels. We also perform in-depth analysis of our manual segmentation to study the annotator error and reliability. Third, a shape-based data augmentation method is invented to provide unimpeachable ground truth for comparison. Segmentation of the MLV images is also performed using a deep learning method, and a set of complexity measures for characterizing the MLV is developed and utilized. And these informatics can also be used to assess segmentation accuracy. So before jumping into the first contribution, which is segmentation, I'd like to briefly describe the data sets that we'll be working with for the remainder of this talk. So on the left is an example from the partial mount data set, which are images of the MLV taken in the superior sagittal sinus. In red are the lymphatic vessels, and the blue showcases the blood vessels. The whole mount data set in the middle image shows the whole sinus connection, so both transverse sinuses from left to right, and the superior sagittal sinus, which extends from the top. And the lymphatic vessels here are shown in green. And the last data set is the spinal data set, showcasing lymphatics present in the spinal meninges, again stained in red. The first contribution is to develop a level set-based segmentation method called Lymphy for accurately segmenting the MLV within a highly cluttered background. As we stated earlier, to begin with, an accurate segmentation of the vessels is necessary for any further analysis. Segmentation here refers to separating the foreground vessels from the background clutter. Although the vessels are stained in only one color, channel separation is unfortunately not good enough to retrieve a good segmentation. Each channel still contains background clutter from the blood vessels and other cells. Shown here is the green channel of the top image, and circled are some bright cells that are still visible within the green channel. The intensity inside of the vessel is also inhomogeneous. We need a segmentation method that can remove background clutter without removing the vessel interior. Thresholding, or keeping only higher intensities in the image, will not work to remove the background clutter in the MLV images. So in this image, the vessel intensity can range from 255, which is the max intensity, to as low as 88, which is a similar intensity to many background regions. And thresholding to remove this background clutter will then remove the interior of the vessel in many locations. The vessel intensity range varies from image to image and even vessel to vessel, so no one threshold would be effective. A traditional method to segment vessels resulting in smooth contours while still capturing fine vessel details is the level set method. The object of interest is represented by the red star. The level set framework defines a curve by the sign distance function phi, where phi equal to 0 is the 0 level set, which moves towards the object boundary. The curve moves according to a velocity v in the normal direction. To move the curve, I move phi according to this equivalent expression. I'll be focusing on the tough formulation designed in 2014 for segmenting tubular structures. The tough or tubularity flow field algorithm uses a vector field to propagate curve evolution axially along the tube. One problem I have faced, however, when using tough for lymphatic vessel images is that the initialization designed to remove the background clutter and noise removes a lot of the vessel content. The segmentation results are highly dependent on initialization, which is basically intensity and scale threshold manipulation. It's difficult to tune the scale initialization in particular because the lymphatic vessels vary widely in size. As shown in the images, the left is the original image, and the right is the tough segmentation result where finer vessel detail is lost due to intensity thresholding and difficulty with tuning the scale parameters. We propose a segmentation method designed for the very specific challenges of lymphatic vessels, and we call it Lymphy, or lymphatic matted phi. It uses a procedure called hierarchical image matting to remove background clutter while harnessing the power of level sets to produce the smooth segmented boundary that we want. The images we have of the MLV share some similarities to fundus images, which are images of the retina. Fundus image segmentation, or matting, separates background from foreground blood vessels. So we try a technique for segmentation of fundus images called matting. The matting procedure begins with a tri-map, or 3-channel image. And one channel is for known background pixels, one is for known foreground pixels, and one is for unknown pixels, U. The unknown pixels are labeled in the second image in red. And the matting process converges when all the unknown pixels U have been labeled as either background or foreground, as in the third image. Using a level set with matting provides the smoothness we desire. On an image, the level set velocity has a magnitude and sign at each pixel. Using the foreground and background created by matting, the level set direction can be set towards foreground objects by changing the velocity sign. At background pixels, the 0 level set should shrink inwards towards objects. At foreground pixels, the 0 level set should grow outwards towards boundaries. This is done by flipping the direction of the level set velocity. The figure shows the process in detail. So at time t, the original velocity field, calculated from the tough forces, is shown by brown arrows. The yellow shape here represents the calculated foreground v from the hierarchical image matting. In A, the red arrows represent an incorrect direction for curve evolution. These arrows are driving feet inside the vessel. These directions are corrected in panel B using the matting label calculated. The now green arrows of the contour evolution velocity have been changed in the normal direction. Lymphy is not just a level set method, it is a matting level set method, the first of its kind. Here are some segmentation results using Lymphy as well as some other segmentation methods. The first column is the original image and hand-segmented ground truth. We compare to L2S or Legendre level sets, a method that deals with intensity variations, as well as hierarchical image matting and tubularity flow field. Lymphy provides the closest match to the ground truth vessel segmentation. We can measure segmentation performance by using the DICE score, which measures segmentation overlap of each method compared to the ground truth or manual annotation. The DICE coefficient ranges from 0 to 1, with 1 being a perfect overlap or match. The results are shown on 3 data sets, the spinal, whole mount, and partial mount. Lymphy has the best performance overall, achieving a DICE coefficient of about 0.7 for the partial mount dataset. The green dataset has the lowest DICE score overall for all methods because of poor channel separation, as we mentioned earlier. Another score we use to assess segmentation performance is the boundary F1 score. This is the harmonic mean of precision and recall of pixels on the segmentation boundary. Again, BF score, just like DICE score, ranges from 0 to 1, and we can see that for all the data sets, Lymphy has a BF score of about 0.8 or above, which indicates a close match with the manual annotation. The last data set is a synthetic data set, which we will discuss during the third objective. One thing to keep in mind, however, is that our comparisons are always performed relative to a possibly flawed manual annotation. It is possible that our automated method can in some cases be better than manual annotation. In the next section, we will show that this is indeed the case for some of our data. So far, we have introduced our novel matting level set method for segmenting the MLV images and shown its high performance with respect to our manual annotation. But what if this manual annotation is flawed? One of the challenges of biological image processing is that manual ground truth is unreliable. We had to overcome this obstacle in order to publish and actually test our algorithm. The ground truth is often overestimating vessel boundaries due to limitations of outlining on a machine. The yellow boundaries in the pictures are often not close enough to the true vessel boundary. As you can see from the leftmost panel, the annotator failed to cut out many small gaps between vessels that are more accurately captured using automated segmentation. To test the performance of manual annotation, 3 annotators, including me, spent many weeks outlining the vessels using image J. The 3 annotations per image were combined using majority voting. Here we are showing the effects of noise on the annotation process. On the left is a consensus segmentation formed using the staple algorithm, which estimates a reference standard from a set of segmentations. We can see the low-level noise left behind by more than one annotator. Majority voting, which we use, removes more of the noise, but is still observed along the boundaries. To assess agreement between annotators, I use kappa statistics. The kappa coefficient is a statistic that is used to measure inter-rater reliability. It is generally thought to be a more robust measure than simple percent agreement calculation shown here, because the kappa takes into account the possibility of that agreement occurring by chance. Fleiss' kappa is used here between all 3 human annotators, as this is the kappa coefficient that can be used between more than 2 observational sets. In red, we see that the Fleiss' kappa between 3 annotators for the partial mount dataset is only 0.62. And all of the values here are only consistent with about moderate agreement. A kappa of at least 0.75 is generally required to consider good agreement, which can be seen in the spinal dataset. However, kappas above 0.9 are generally preferred, which we cannot show in any of these datasets. In the second table, we show Cohen's kappa and percent agreement between segmentation methods and our manual annotation. I'd like to point out the kappa value of 0.7 shown in green between Lymphy and the annotation of the partial mount dataset. For this dataset, the kappa statistic shows that agreement between Lymphy and annotators outperforms inter-annotator agreement. For these images, Lymphy is superior to manual annotation. The staple algorithm, in addition to estimating a consensus segmentation, has been widely applied to compare the performance of different algorithms and experts. In this table, sensitivity or true positive rate and specificity or true negative rate is shown between each annotator and Lymphy with a consensus segmentation. True positive rate here means the marking of a pixel as foreground by the annotator that correctly matches the consensus segmentation created by the staple algorithm. Green here represents better performance by Lymphy than at least one annotator, and red for very poor performance by a single annotator. I would like to point out the value of 0.56 sensitivity shown by a single annotator for the whole mount dataset. Clearly, relying on a single annotator for segmentation is very risky, and Lymphy's performance in detecting the vessel foreground does not drop to this level for any of the data sets we used. To conclude this section, we show a failure to annotate the boundary accurately by manual segmentation. The top left shows the original image of a lymphatic vessel with our consensus manual segmentation on the right. Using Lymphy, shown at the bottom, we can more closely match the true vessel boundary. In this section, we have shown that manual annotation is unreliable to use as ground truth. With the next objective, we will address this issue by generating synthetic data where the truth is known. The third contribution is to build a shape-based data augmentation method that generates reliable ground truth. If we could generate realistic lymph images where the boundary is known, we can dramatically advance image analysis research for lymph in terms of our research and research in the wider community. We propose using elastic deformation of existing vessel data into new data, and we will perform this deformation using shape analysis tools. First, we need to move from the images that we have to the shape domain. So the first step is to grab all individual vessels from our annotated images. Each individual vessel is represented as a shape, and the holes inside the vessel are kept aside. Taking random pairs of the vessels, interpolation is performed to achieve a vessel morphing. The reserved holes and gaps are also deformed using deformation transport. These are then placed within the morphed vessel to result in binary single vessels. Using our majority voted ground truth from our annotations, as we described earlier, the individual vessels are gathered. So what is a shape? A shape is a representation of an object that only includes its outline. The shape contains no texture or color information. Shown are just some examples of shapes. To reparameterize the object boundary beta of the vessel, we use the square root velocity function, or SRVF. The curve beta is sampled, and the SRV function is used at each sampled point to represent the shape. The representation is a matrix Q, which embeds the shape information in a shape space. When we use the SRVF, there are features such as geodesic path, distance, and interpolation between shapes that we can use. The SRVF also accounts for all rotation and scale variance of a particular shape. To create a new vessel, morphing is performed between two vessel shapes on the shape space. I chose to use morphing between vessels so that I don't have to assume a distribution of the existing shapes. For a data set of just 18 images, there can be about 450 original vessels, which results in over 100,000 vessel pairs. And multiple morphed vessels can be formed from each vessel pair. So using these possible combinations of vessel pairs, almost infinite images could be generated. If we sample a geodesic path between two different shapes on the shape space, we will find the morphed shapes in between. This figure is an example of interpolated vessel shapes along a geodesic path from vessel 1 to vessel 2. Sampling with step size delta between the two given vessel shapes provides the new vessel shapes, which we will be using as synthetic data. The representation Q1 and Q2 of the two shapes need to have the same dimensions. For a 2D shape, the size of Q is n cross 2, where n is the number of samples taken to characterize the vessel boundary. Because the path taken between two SRVF defined shapes is Euclidean, I take convex combinations of the two Q matrices in order to interpolate. So the shape only keeps the outline of an object, as we discussed earlier. But to recreate a lymphatic vessel, the holes are an important characteristic. So I represent the holes as their own shapes. Using the equivalent shooting direction and amount, or velocity, used to move from vessel 1 to vessel 2 in the top panel, we morph the holes that were within the original vessel. Essentially, we apply the same amount of bending and stretching on a new shape, in this case, a vessel hole. The output from this workflow are individual binary vessels with holes fitted proportionally according to the vessel size. To generate the full synthetic images, we select foreground vessels from the binary morphed vessels we just created, and these will serve as our ground truth. The foreground signal is then attenuated using a Gaussian filter to better match the original images. Background clutter is also created from the morphed vessels, but set at a lower intensity. We combine the foreground and background layers, add Gaussian blur and Poisson noise, which are characteristics common to confocal microscopy images. The final results of the workflow described are grayscale lymphatic vessel images. The grayscale image is displayed in a red color map in the bottom right corner to better show the background noise. The direction of the vessels is based on the position of the original vessel in the microscopy image. As these synthetic images were generated from the partial mount images, the vessels are simulated to grow laterally along the blood vessels in the sinus. These histograms of a near constant intensity region in both a microscopy image and a synthetic image show that we added noise similar to that in the real images. The distribution and range of intensities of the background regions are similar. The region from the real image is a little larger, and that's why the frequency or y-axis is higher. Here we show the regions from which the histograms were created. As mentioned, the intensity ranges are similar. Some clutter is present in the microscopy image, however, that was not simulated in the synthetic image. Using our synthetic images with built-in ground truth and segmenting using Lymphy, we achieve much higher dice coefficients. In this case, the match in results is almost perfect with a dice coefficient of 0.98. Here are the average dice scores for all the methods tested on the synthetic dataset across the bottom row in green. The average dice coefficient using Lymphy is 0.89, showing good performance on the noisy, varied intensity images. Performance by Tough is extremely low because all the background clutter is included, and noise is also emphasized. In this section, we have shown our method for generating synthetic MLV images complete with accurate ground truth labels. Lymphy performs highly on this dataset and better than other methods on the other datasets, even though the manual annotation we compare to has flaws and does not represent ground truth. In the next section, we will explore the possibilities of deep learning for MLV segmentation. Now that we have seen the efficacy of our level set segmentation method, Lymphy, let's look into a study on how deep learning performs on the segmentation challenge for MLV. Segmentation can also be viewed as a classification problem, where we need to classify each pixel as either a foreground pixel or a background pixel. We will use a convolutional neural network, or CNN, to classify the different pixels. We are using a CNN because it is shown to work well on biological images, and there are a number of CNNs designed specifically for segmentation of biological data, such as the UNet. The creators of the UNet built a CNN for segmenting cells in biological microscopy images. The downward portion of the U includes downsamplers, and the upward portion is an expander network, or upsampling part, before the output layer to increase the resolution of the segmentation output, thus creating the U shape in the network architecture. Using augmented data and the trained UNet, segmentation results have vastly improved compared to other competing methods in the literature. Variations of the original UNet have shown great success on segmentation of blood vessels in fundus images, which again are images of the retina. We use one such implementation of the UNet to train and test using our lymphatic images, as they also contain vasculature. So we have 39 microscopy images in total. These original 39 images were split into 30 training and 9 test. Current approaches to data augmentation include simple addition of noise, flipping, cropping, and shifting. And these are the techniques used in the original UNet framework, so we use this data augmentation approach as well. We crop the ground truth labels at the same time as cropping the original images for augmentation. We ran our deformation pipeline to generate 999 images of size 512 by 512 to act as training and testing for the ground truth masks and original images. The smaller image size is necessary, otherwise we don't have enough data to train the network. Once the data had been augmented, the augmented images were split into 750 training and 249 for testing. So, to summarize, 750 augmented images were created from the 30 original training images, and vice versa. Here we show results on a cropped spinal lymphatic image with other state-of-the-art segmentation methods. The original red channel image of lymphatics is shown in the far left corner. The majority voted ground truth is shown on the left top panel. Chan-Vesey, L2S, and TUF are all level set segmentation methods. Hierarchical image matting, as we discussed before, is in the bottom right corner, and it's a correlation-based segmentation method for the segmentation of retinal images. The UNet output is actually a confidence map, which we threshold at confidence level 0.5 or 50% to get a binary segmentation result. Using UNet provides a dice score of 0.92, a very high dice score in its own right, and much higher than all the other competing methods. This is a high segmentation result, as the highest value the dice coefficient can take is 1, which indicates a perfect match with the ground truth. The details in the UNet segmentation result are much clearer, even though the staining in the original image is very weak between branches of the vessel. The overall Dice coefficients for all methods tested are shown here. Most of the Dice coefficients for the test data results segmented by unit are between 0.7 to 0.9. The average, mode, and median Dice score are all higher using UNet than any of the other methods. There is also less or equal spread in the results compared to other methods, so most of the scores are compacted around the average. This means that the segmentation is more accurate compared to the ground truth. This makes sense, as the UNet method is the only method shown here that is actually trained with the ground truth in mind. Accuracy in segmenting the vessel is extremely important, as it will affect the complexity measures we wish to calculate in the next section. For segmenting the full-size MLV images, Lymphy is still the preferred method. However, this study on the cropped MLV images shows great promise for future work using deep learning. Now that segmentation is in hand, the fifth contribution is to develop a set of informatics for characterizing the MLV. These measures can also be used to assess segmentation accuracy. Some measures currently taken on the vessels are area in microns and average width. And some other measures also include number of sprouts or branches, length, etc. The width is sampled at various points along the vessel manually. Width measurements vary wildly, up to 30%, because different users sample the vessels at different points. Average width also blurs over stark differences in lymphatic size across a network. Our goal is to create automated, reliable measures to quantify and characterize the meningeal lymphatic vasculature. We can first build a skeleton from the segmentation to start the automated complexity analysis. Skeletons are 1 pixel wide backbones of image objects. Displayed is a lymphatic vessel skeleton with endpoints marked in green and branch points marked in red. One measure of interest for lymphatic vessels is vessel length, which can measure lymphatic regression. Lymphatic regression is where the vessel grows shorter in length over time, usually with age. The vessel length is calculated as the total number of pixels along the skeleton, as the skeleton is 1 pixel wide. To develop a different complexity measure, we draw inspiration from the ramification index used to study microglia. The ramification index is defined as the ratio of the perimeter and area of a glia cell normalized by the same ratio for a circle of the same area. This measures how spread out or ramified the processes or branches of the microglia are. Since the lymphatic vessels are not centered at a cell body or soma, it doesn't make sense to compare to a circle. Instead of comparing to a circle of the same area, I decided to compare to a simpler vessel, a vessel with one smooth contour all along the outside. A rough approximation of the boundary is shown on the left with the boundary in yellow. The boundary is then filled to create a simplified vessel, whose perimeter and area are calculated to compare to the original unfilled vessel. The new ramification index formula is then a ratio of the perimeter and area of the two vessels. Another similar measure we developed is inspired by porosity from materials. Porosity, or void fraction, is a measure of the void or empty spaces in a material. It is the fraction of the volume of voids over the total volume. Porosity is always a number between 0 and 1, unless it's described as a percentage. A material has high porosity if it contains large spaces, such as the figure on the left. Vessel porosity is calculated as the area of capillary loops, which are the gaps where a vessel branches and reconnects to the larger network, over the total surface area of the vessel. We use area instead of volume because our images are 2D. Pictured are some images of the capillary loops as shapes. Vessel porosity is similar to ramification index in that it measures how much space is filled within the larger vessel boundary. Here are some complexity results. On the left is a simpler vessel with lower vessel ramification index. A more complex vessel is shown on the right with a higher vessel ramification index. The new RI is able to discriminate distinctively between simpler and more complex vessels. The original ramification index for microglia scales similarly with the two vessel types, which tells us that the vessel ramification index is measuring relevant complexity information. While the previous RI for microglia also is discriminative between the two vessel types, the new one I developed makes more sense for this problem. All three metrics, length, vessel ramification index, and porosity, were run on segmentation results and compared to the ground truth using mean squared error. The unit segmentation results have the lowest error for all three measures compared to the ground truth complexity values, as shown in the table. Vessel length varies widely because the skeleton produced changes enormously with any small change in the segmentation. And as porosity is always a value between 0 and 1, the MSE for porosity is lowest across all the methods. To recap this section, informatics were developed specifically for the MLV, including two new measures, vessel ramification index and vessel porosity, and these were also used to assess segmentation performance. The contributions of this thesis are a novel matting-based level set segmentation method that accurately segments cluttered vessel images, analysis of the manual segmentation revealing problematic errors and relying on manual annotation as ground truth, as well as providing a data set of 39 consensus annotations of MLV images, a data augmentation method based on shape deformation that can be used on any shapes where elastic deformation is reasonable, testing the efficacy of deep learning for MLV segmentation with promising results in terms of a high dice coefficient, and creating new complexity measures to analyze lymphatic structure and using these measures to evaluate segmentation accuracy. This work opens a gateway into future work on lymphatic bioinformatics. In conclusion, I would like to outline some future directions for this work. Our shape-based deformation method can be used to model age-related or disease-related deformation of the meningeal lymphatic vessels. The shape deformation can also be used in conjunction with conditional generative adversarial networks in order to generate even more realistic MLV images. This new synthetic data could then be used for CNN training. And finally, using the MLV skeleton we generate, a graph structure could be created specific for the MLV that accounts for self-loops in order to bring the power of graph signal processing to MLV analysis. These are my publications. And I would like to thank my committee members for being here today, especially to my awesome advisor, Dr. Scott Acton, and my committee chair, Dr. Zhongli Lin. Thanks also to my neuroscience collaborators for making this work possible. Dr. Yasmin Hertz was formerly on my committee, but due to her new position at the Washington University School of Medicine is now a special guest. Thank you very much to Dr. Barry Condren for stepping up to fill her place on my committee. I would like to also take a moment to remember our dear departed Dr. V, my previous committee chair, and also my teaching mentor. I wouldn't be here today without her support, and I will always miss her. Special thanks to Viva and Vital Lab members for their advice and inspiration for this work, as well as for becoming some of my dearest friends and colleagues. And finally, thank you to my loving family, especially my mother, my father, my brother, and my husband, as well as all of my lovely friends for your constant love, prayers, and encouragement. And I'll be happy to take any questions.